Okay, part 5, slide 24. We can look at, we'll go back to the synthesis aspect of the virus, and we'll take a closer look at our different kinds of viruses and how they make the copies of themselves. DNA viruses, RNA viruses, we'll look at the difference between positive and negative sense strands, and we'll look at reverse transcribing viruses. DNA viruses, they replicate using the host cell machinery. Cells themselves are based upon DNA, so it's kind of an easy job for DNA viruses because all of our machines, our ribosomes and our RNA polymerase, those are all built around handling DNA. When we come to RNA viruses, the virus has to do a little bit extra. It has to replicate some molecules that are not present in cells. When we come to reverse transcribing viruses, again, this is kind of probably the most convoluted of the methods. We'll look at this thing called reverse transcriptase and also the integration of DNA into host genomes. So we are on page 346 and 347, which are a couple of very intimidating pages in the textbook. And let's start with DNA viruses. And there are two kinds of DNA viruses. There are double-stranded DNA viruses designated by DS, and there are single-stranded DNA viruses designated by SS. Double-stranded DNA viruses, super straightforward because cells already work with double-stranded DNA. So RNA polymerase is ready to go to read double-stranded DNA and make messenger RNA. And then ribosomes are ready to go to read that messenger RNA and make the proteins. We'll look at down here that the enzyme that is responsible for copying DNA is DNA polymerase. DNA viruses often bring their own because a cell isn't actively replicating a lot of the time. A cell's RNA polymerases and ribosomes are always on. A cell is always building new proteins, but only during cell division is DNA polymerase active. So if the virus wants to make copies of its genome, it doesn't want to have to wait around for the cell. It'll just bring the molecule with it, and it will replicate its genome with its own DNA polymerase. In terms of single-stranded DNA, the only thing they have to do is they have to become double-stranded first, which means they have to build the second part, the complementary strand, using cell machinery, and then they can be read and transcribed and translated, the genes into proteins. Replication of the genome occurs in the nucleus because that's where we have all of our stuff. So that's where we have all of our DNA nucleotides, our A's, T's, G's and C's, the same A's, T's, G's and C's in our DNA viruses. So whilst the DNA viruses may bring their own polymerases, they are stealing all of our resources. They're stealing our T's and our A's and G's and C's. They're stealing our nucleotides to build their own genomes. There are some exceptions. Pox viruses replicate in the cytoplasm because they bring with them all the stuff they need. They don't rely on the host cell for any of the machines. They only rely on the host cell for the component parts. on to RNA viruses. The majority of RNA viruses are single-stranded. They mostly replicate in the cytoplasm. They require a molecule called replicase. Replicase is a special kind of RNA polymerase. So RNA polymerase, as we know it in cells, reads DNA and makes an RNA copy. So it reads the DNA of our genes and makes messenger RNA that then goes on to be translated into proteins. In this case, we are starting with RNA, and we want to make RNA. So we need an RNA-dependent RNA polymerase, and that does not exist inside cells because no cell ever needs to do that itself. So either the virus brings this replicase enzyme with it inside the capsid, or it has a gene in its genome that is transcribed and translated, in this case just translated, into the replicase. One thing about replicase, the replicase enzyme, is it lacks what we call proofreading, which means it makes lots of mistakes. So we have, in this case, so replicase lacking proofreading. Remember our bases of RNA, A's, U's, C's and G's. Here's our RNA genome, and we would expect that to be replicated A to U, U to A, C to G, and G to C. But because of our replicase lacking proofreading, it does make mistakes, just like the mistake I made. It can, and then this goes into A, U, G, C, and you see that the replicated genome is now different to the genome in which we started. So mistakes can easily get introduced into the genome because of the mistake, the mechanism of replicase making mistakes. And if this was a segment of a gene, and this gene built a spike protein, and this lettering built a circular spike protein, but through the mistakes made by replicase, we now build a different shape of spike protein. We now have a virus that has a different spike, and that then may be able to interact with a different receptor. This change may be very gradual, from generation to generation to generation. And this process of slowly changing the surface structure of the virus particle is known as antigenic drift. Mistakes made in genome copying, known as mutations, those mistakes happen in genes that code for particularly surface proteins, in particular things like spike proteins, called antigenic drift because it also affects the ability of our immune system to attack the virus. So here's our virus. Here's the original virus. Here's the virus capsid. Here is the spike protein, and currently, originally, the virus had a circle spike protein. And our body, having seen this virus, would have mounted an immune response, and part of that immune response would be an antibody production. And here's our Y-shaped antibody, and here's the variable region of our antibody, and you can see that our variable region of the antibody can recognize the circle of our virus. But here's our new virus after the mistake, after the error from our replicase, and now our antibody can no longer stick. This shape of our antibody variable site is no longer a match for the shape on the virus spike. This is one reason why, for example, influenza keeps changing year to year, and while we have to keep updating our immunization. We can't have a single shot against influenza, because next year the antibodies that we have built up may no longer match the antigens on the surface of the virus. So antigenic drift is an important mechanism of viruses gradually changing the shapes of their antigens, i.e. the proteins on their surfaces. And drift, kind of think like drifting, it's a slow process. Typically, it's thought of as a slow process.